Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting, and I have another great lesson ready for y'all today. So let's go ahead and dive into it, and let's try to learn something while we're at it. So today we're talking about osmosis. Osmosis is very similar to diffusion, but there's one key difference you have to remember. So what is osmosis? Osmosis is the movement of water. That's very important to remember. Anytime water is moving around, the answer that they're looking for is osmosis. And here's a little tip to remember that. Let's think of the molecular formula for water. What is the molecular formula for water? H2O, right? 2O is what we want to focus on. Because if you look at the word osmosis, it has two O's in it, right, when you spell it out. So to me, that's an easy way to remember what is the movement of water? It's osmosis. H2O, osmosis has two O's. So that's just a little key to success you can remember to get this stuff under your belt, okay? So in osmosis, what's going on here is that the water moves across a membrane, just like diffusion, from an area of low concentration to an area of high concentration of solutes, okay? So what's going on here is that the water wants to even out the amount of solutes that are happening. This is a type of passive transport, so there's no energy required here, okay? Let's remember that water in osmosis, we're moving across a cell membrane, and cell membranes are semi-permeable, which means that they let some things in and out, but not everything. So sometimes in a semi-permeable membrane, water might be able to move through it, but the sugar might not be able to. So in our last lesson, we were talking about the solutes moving. Now we're talking about the solvents moving. The water is going to be the thing moving around here. Okay, so let's go ahead and get into it and see if we can figure out what's going on. So again, osmosis is a type of passive transport. If it's passive transport, are we using any energy here? We are not. Passive transport does not require energy. Okay, osmosis, again, is the diffusion of water across a membrane. Remember that. If water's moving, it's going to be osmosis. This is going to be very important for maintaining cell homeostasis, and I'll get into that in a little bit later on. Okay, and water moves from high concentration to low concentration of water. Okay, so it's kind of confusing because it's doing sort of an opposite thing here. So when you're talking about the solutes, it's moving from low to high. But when you're talking about water or the solvent, it's moving from high to low. So let's take a look at this picture and I'll kind of break it down for you, okay? So looking at this picture, we have a tub of water, and in the middle there's a barrier, that's the membrane. Okay, well, where are all the solutes here? The solutes are on the left-hand side. Those are the orange dots. So there's more solutes on the left, and there's less solutes on the right. In this example, the solutes can't move. The membrane is stopping them from moving. So what's going to happen is some of the water on the right-hand side is going to flow into the left-hand side to balance it out, okay? So it's moving from where there's a lot of water to where there's not as much water relative to the amount of solutes in there, okay? So here's another way to kind of look at that picture, okay? So what's going on here, remember osmosis is controlled by the amount of solutes on either side of the membrane. So we have this kind of horseshoe-shaped tube, and there's a membrane down the middle. The membrane is going to let water move across it. Water can move through things really well, but the solutes aren't always going to be able to get across that membrane. So what's happening here is that there are more solutes on the right-hand side. There's more solutes over here, more little dots, and there's less solutes over here. So if you let this sit for a while, what's going to happen is that water starts to move over to the right-hand side. And what is going to be the end result is that there's less water on the left and more water on the right, because water always moves towards high solute. That's what you really want to remember. Water always moves towards high solute, okay? And so this would be an example of how that water is following the solutes. Because there's more solutes on the right, the water is going to move over that way, and you're left with less water on this side of the tube and more water on that side of the tube. Great. Okay, so I wanted to talk about this in reference to our bodies and our cells, okay? And again, this is dealing with osmosis and what's happening to the water. So I have a question. What if there is more solute outside of the cell than inside? Well, remember that water always moves towards high solute. So what would happen here is that if there's more solute outside, the water is going to leave the cell. If water is leaving the cell, that's going to cause the cell to shrivel and to shrink. And an example of this is like pouring salt on a slug. If you pour salt on that slug, the slug is real slimy. It's got a lot of water in it. But if you pour salt on it, all of that water is going to leave the body and moves towards the salt, and that's what ends up kind of, killing the slug, is that it loses all of its water. Okay? So this causes the cell to shrink, because if the water's moving out of it, it's like letting the air out of a balloon, right? It's going to start to kind of shrink a little bit. Okay, so what if the opposite happens? What if there's less solute outside of the cell? Less solute outside of the cell, more solute inside of the cell. Okay, in this case, the opposite's going to happen. Because there's more solute inside of the cell, the water's going to rush into the cell, and that's going to make the cell expand. It's like if you took that balloon and blew a whole bunch of more air into it, it would get a lot bigger, right? The same thing's going on here. Because there's more solute in the cell, the water's going to rush in, and that makes the cell expand and get a little bit bigger, okay? In fact, in a bad case scenario, this cell could get so large that it would actually burst, okay? And we'll talk more about that next time, okay? So this is really the environment that we want. This is something a little bit different. What if the amount of solute is the same from the inside and the outside of the cell? Well, the water's not going to stay put. Remember that in dynamic equilibrium, stuff is still moving around, but the overall size of the cell is not going to change. So the water's moving into the cell, the water's moving out of the cell, but it's happening at the same rate, and so that would cause the cell to not change. This is what our cells are doing most of the time, every day, even right now, is that it's taking in some of the water that you've had in your diet, it's getting rid of some of that water, and it's just kind of cycling it. Okay, so let's look at this picture and kind of break it down a little bit. Okay, over here on the left, and the leftmost example, this is like our slug and the salt situation. If you pour that salt on the slug, there's more solutes outside of the cell, and so you can see in the bottom on the left-hand side, all of that water is going to leave the cell. If the water's leaving the cell, it's going to shrivel like a balloon that you're letting the air out of. Okay, in the middle, this is the situation that your blood cells are in right now. Is an equal amount of solutes inside and outside, and so you can see that there's two arrows here. Some water's coming in, some water's going out, but they're in an equal amount, so the overall size of the cell is not going to change. And then in the last picture way over there on the right, this would be if there's more solutes inside of your cells. If there's more solutes inside of the cells, the water's going to come rushing in. When it comes rushing in, that's going to make the cell swell up, and you can even see that there's one little example right here where the cell is actually bursting, and that can be pretty bad. That's why this can kind of be a dangerous scenario, and I'll give you some examples of that later on. Okay, great, y'all. So I briefly just introduced a couple of these, but I have some review questions I'd love for you to work on. So why don't y'all go ahead, pause this video. Teacher, go ahead and pass out this worksheet to your students. Y'all take 5 or 10 minutes and work on it by yourselves, and then when you're ready for a review, come back to me, unpause the video, and we'll work through this part together, okay? So go ahead, pause the video, and then come back when you're ready. All right, y'all, I know this gave y'all no trouble at all, but just in case somebody's a little bit confused, let's make sure we review it together, and then that way everybody's on the same page. So what we're doing here is it's giving us a couple of different examples, and we want to use either the words osmosis or diffusion. So if we think that an example of diffusion is taking place, we'll write that, and we'll write osmosis if we think that that's what's happening. Just a little hint here, remember what I told you about osmosis. What is moving in osmosis? It's water. So if we ever see water moving around, we're going to go with osmosis. Anything else will probably be diffusion. So let's go ahead and hop into it and see what everybody got. So the first one here says, the student sitting next to you just came from gym class and forgot to shower and you can tell, okay? So imagine a student comes in, they could even be sitting on the opposite side of the room. So the smell is kind of coming from them, but how did it get all the way over to you? Well, that's an example of diffusion. So let's go ahead and write that one in, all right? So this first one would definitely be a form of diffusion. Would definitely be a form of diffusion, okay? I'm going to write either the words D or O for these, okay? That'll be a little bit easier for me writing on this board. So the next one, after sitting in the bathtub for hours, your finger starts to look like prunes. Why do your fingers start to get pruny if you're sitting in the water? Well, actually, surprisingly, that's an evolutionary thing. When you're in water for a while, you don't really have much of a grip, but when your fingers start to look like prunes, they get all those folds in there, and you actually get a little bit better of a grip underwater. So that's why your fingers get pruny. But in this example, it's water coming into your cells. So if water's moving, that's going to be osmosis. And again, I'm just going to write the little letter O there. That'll be a little bit easier on this smart board. Okay, the next one says, the girl sitting 2 rows ahead of you put on too much perfume this morning and now you're smelling it every time you take a breath. Well, are we dealing with water at all? Not really, so this is going to be diffusion, okay? That's right, let's go to the next one. Letter D. One way to get rid of slugs in your garden is to sprinkle salt on them so they shrivel up. Well, if you put salt on them, if they shrivel up, it's because water's leaving their body, and so that would be an example of osmosis, okay? So the next one, this says, yum, something smells good. The neighbors are cooking on the grill. So they're cooking way in their yard, but the smell's getting all the way to you. Why are you able to smell that? Well, those smell particles are diffusing. They're diffusing, okay? So D for that one, all right? Next, gargling with salt water when you have a sore throat causes your swollen throat cells to shrink and feel better. So what's going on there? If they're shriveling, that's probably water leaving, right? Well, if that's water leaving, that's going to be osmosis. Great. All right, and then here's the next one. Oxygen molecules move from the air sacs in the lungs across the cell membranes into the blood. Well, there's really no water moving here, so this is going to be diffusion. And oxygen moving in and out of your lung sacs like that is a great example of diffusion. Okay, great work. I got a couple more here for us. Let's see what y'all came up with. Okay, so this says use arrows to indicate the direction of diffusion in each case below. So this is very similar to something we were looking at last time. Remember, we're really going to draw 2 arrows here. We're going to draw a big arrow to show where most of the solutes are going, and then that second little arrow just is to remind us that even when they get to that equilibrium, they're still kind of moving around. Okay? So letter A, well, we're moving from high concentration to low concentration. So where is there a higher concentration? It's inside the cell, and so mostly they're going to be moving out. They're going to move out of the cell a little bit, okay? So I'm drawing a bigger arrow pointing outwards. Now, just because most of them are moving out doesn't mean that none of them are moving in, so there is going to be a little bit of diffusion into the cell, but most of it is moving out. That's why we have the bigger arrow there. Let's look at the next one, B. All right, so we're moving from high to low concentration. Where's the high concentration? There's more solutes outside of the rectangle and there's less inside. So overall, the majority of our movement is going to be into the cell, okay? And then just a little bit will be outside of the cell once we reach that dynamic equilibrium. Let's keep it going. Number 3. For each of the situations below, use arrows to indicate the net movement of sugar into or out of the cell. Now with these, we could draw 2 arrows like we have in the past, but this says net, so it's really just asking us for the overall change. So here I think really either one is good. We could do 2 arrows or 1. I'm just going to do 1 on these because it is saying the net, so that's the overall change. Okay, so for each of the situations below, use an arrow to indicate the net movement of sugar into or out of the cell. Okay, assume that the sugar molecules can pass through the cell membrane in each case. All right, so they're kind of cut off from my slide a little bit. You should see them just fine on your sheet. I'll go ahead and write them here. This says 1% inside and 5% on the outside. Okay, so let me write that there. 5% on the outside. This one is saying 3% on the inside, 1% on the outside, and then 1% here as well. Okay, so that's going to be the values we're using here. All right, well, if there's, on our first example on the leftmost side, if there's 1% sugar on the inside and 5% on the outside, the net movement of the sugar is going to be outside the cell, because that's where, I'm sorry, I got that reversed. The net movement is going to be inside the cell because we're going from high to low concentration. There's more on the outside, so they're going to move to where there's not as many. So they're going to be moving it. Okay? The next one, there's 3% on the inside, 1% on the outside. We're going from high to low, so the net change is going to be outside of the cell, right? We're going from high is 3% to low is 1%. Now, this one is a little tricky. It says 1% on both, so this is actually equal. So there is no net movement. So here I think it is okay to draw two arrows, okay? Because some is coming in and some is going to be moving out, okay? Great work, y'all. Great work. Okay, I got a couple more here, so y'all hang in there with me and let's see what we got going on here. Okay. So now I've got some letters on the right-hand side and some terms on the left-hand side, and we're going to see if we can match them together. Okay, so let's look at this. So the first one says concentration. Okay, well, which of those letters can we use to represent concentration? Well, remember that concentration is the amount of substance in a certain place. So I think B is going to be great for this one. Okay, remember a high concentration is you got a lot of solutes, and a low concentration is you don't have as many of those solutes. Okay, 7 is diffusion. Well, what's happening in diffusion? Okay, well, this is how particles like oxygen get into and out of our cells. Okay, it's that movement from high concentration to low concentration. Okay, the next one says equal amount of water inside a cell as outside. Okay, I'll give you a hint. It's going to be one of these three pictures at the bottom. Which of those do you think represent equal amount of water in and outside of the cell? I'm thinking letter E for this one, okay? Because you can see some water's moving in, some water's moving out. The arrows are the same size, so it's in an equal amount, right? Great. Let's keep it going. 9 says more water outside a cell than inside. So which of those pictures down at the bottom do we think represents more water outside of the cell? Okay, well, I'm thinking letter G for this one. If there's more water outside of the cell, then the water's going to be rushing into our cells and it's going to swell it up a little bit. Okay, 10 is asking for osmosis. Do we see a good definition for osmosis here? Remember that it's moving water. That's really important, okay? So I see moves water. I know right away that that's the answer. So this one's going to be C. All right, 11 says more solutes outside a cell than inside. So if there's more solutes on the outside, that means the water is going to leave our cells. So for this one, it's going to be letter F. You can see the water is leaving the cells here, right? So that's going to be letter F for 11. And then last one, selectively permeable membrane. That is going to be letter D because it allows some substances through, but not everything. That's what selectively permeable means. Selectively means like you're picking what you want, and then permeable is the ability to let things in and out. So it's picking what comes in and out of the cells. Okay, y'all, great work. I got a little bit more for us. So let's see what's on the next slide, and we'll knock that out. Okay, so what we're doing here, this says complete the table by writing whether the solutes and water move inside or outside of the cell. It gives us a hint. With diffusion, solutes move from an area of high concentration to low concentration, and with osmosis, wherever more salt is, the water follows. So with water, it's always going towards high solute. Okay, To me, I think it's going to be a little bit easier if we do them by columns. So let's do all of the diffusion ones first, and then we'll come back and do all of the osmosis ones next. So looking at the diffusion one here, okay, we've got 5% inside the cell and 10% outside of the cell. So where is the solute going to move? Well, remember that we always move from an area of high to low concentration. There's more on the outside than on the inside, and so that means that overall we're going to be moving into the cell, okay? Great. All right, the next one says 10% salt inside the cell and 10% salt outside of the cell. So these are actually going to be equal. So for this, I'm going to write both. Okay? Remember that in that dynamic equilibrium, they're going to be moving into and out of the cell. Okay? Both here. Great. All right, so now we got 3% glucose on the inside and 1% on the outside. We're moving from high to low, so the overall here is going to be, it's mostly moving out, okay? All right, now we got 2% protein inside the cell, 1% on the outside of the cell, so the overall change here is going to be out again. Okay? Here's the next one. It's another equal one, 9%, 9%, and so this is going to be both because they're still going to be moving even though they're equal. Okay? Both. All right. Here's our last one for the diffusion. 13% water on the inside, 25% water on the outside. We're always going to move from high to low, so this is going to be, we're moving outside of the cell, okay? Great work. Okay, so those are all the diffusions. Let's talk about osmosis. Remember with osmosis, it's always moving towards high solute. So we want to find where the high solute is. The water's always going to move in that direction. All right, so for the first one, where is there more solute? There's more solute outside of the cell. So that means that the water's going to move out. Okay? All right, how about the next one? 10 and 10. Those are equal. So the water is still going to be moving here, but it's moving in and out at the same rate. So let's write both again for that one. All right, the next one, we're going to high solute. There's 3% on the inside, 1% on the outside, glucose. So that means that the water is going to be moving in to this cell, okay? Because that's where the high solute is. All right, 2% on the inside, 1% on the outside. That means we're moving in again, okay? Here's the next one, 9% salt inside and outside the cell. They're actually equal. The water's still moving in both directions though, so let's go with both. And then lastly, we got 13% on the inside, 25% on the outside, okay? So we want to move towards high solute. The high solute is outside of the cell, so this one, okay, let's see, we're moving towards, okay, so there's actually more, this one's a little tricky because it's talking about water. So with the water, we're trying to even them out a little bit. So for this, it's actually going to be moving in because there's less water and there's more water on the outside. So this one's actually going to be moving in, okay? Y'all, great work. Y'all are so smart. Thank you so much for hanging in there with me. We got a couple more assignments we'd love for you to practice this stuff on. So make sure to give that your best effort and come back here next time. I hope y'all have a great day.